In developmental networks, specific patterns are present that determine whether a gene is expressed or inhibited. In this example, we have a thorny dragon, and the network we are analyzing is S promotes P, which inhibits I, which inhibits K, which promotes E, which will promote the expression of spikes. This is the wild type variant, where no mutations are found. With enough E protein present, cells of the skin are able to create spikes, but not all of them. This will be explained later. Mutations in any of the proteins will lead to an inability for that protein to act on the next protein downstream. For example, a mutation in the P protein will prevent I from being repressed, leading to a buildup of I, which causes K to become repressed, not allowing it to promote E, which will lead to no spikes. If I is mutated, K will build up and more effectively promote E, leading to spikes. A mutation in I and E will firstly cause K to build up, but with no functioning E for K to affect, no E proteins are created and no spikes are made. Finally, if S and K are mutated, P will not accumulate, so I will build up, which represses K, but with no functioning K present, it doesn't even matter. E will not be activated and spikes will not form. Now, let's rewind it to where I said cells of the skin are able to form spikes, but not all of them. What exactly did I mean? Well, a DNA regulating process called DNA methylation occurs. DNA methyltransferases heavily methylate certain regions of the DNA. This attracts deacetylases, enzymes that tighten histones to DNA, stopping DNA from being read, which essentially silences that DNA from being transcribed and causes no proteins to be made. This does not occur with DNA strands that are not wrapped around histones, explaining why some cells express spikes while others don't. DNA not wrapped around histones will be easier to transcribe, allowing the developmental network to be expressed. Methylated DNA wrapped around deacetylated histones will be difficult to read, leading to no transcription and no expression of developmental networks. No transcription, no spike proteins, no gene expression, no spikes. Sorry, little buddy. And these are the references.